Okay, so we're going to talk about excitation-contraction coupling. For the most part, like I said, this is review, but we're going to go with it. So, one of the things that we're going to talk more about that didn't really talk about in my human phys class is this idea of calcium reuptake. Okay, so if we look here, we've got a neuromuscular junction, and there's a lot of things going on here that are important to know. Okay, so we've got the axon terminal, right, which is the end of the neuron. We've got these vesicles, okay? The little dots are the acetylcholine. We've got these enzymes called acetylcholinesterase, which is an enzyme that breaks down acetylcholine, okay? And we've got our muscle cell. We've got some ligand-gated or chemically gated ion channels, right? They're waiting for acetylcholine so they can let sodium in. And then we've got these voltage-gated sodium channels, which will ultimately let more sodium in and cause an action potential. Okay, so quick review of how this works. An action potential will come down the axon terminal. Okay, you're going to have calcium gates open, so that's right here. So calcium gates open. We got them on both sides. Calcium will go in because there's more on the outside than the inside. That causes exocytosis, so these vesicles move to the surface where they release acetylcholine. Acetylcholine will then bind to these chemically gated or ligand-gated sodium channels. So sodium will go in because there's more sodium inside than outside. Okay, and that will activate these voltage-gated channels, which will let even more sodium in, and it causes an action potential. Okay, and so we've got most of that here. So we basically got to this part right here. Okay, and then my picture doesn't really show it. I've got another one that's coming up here in just a minute that will. But what happens is that action potential goes down along the membrane and into the T-tubule, where it activates a voltage-gated calcium channel called a DHP receptor, which lets calcium into the sarcoplasm. At the same time, when that DHP receptor is activated, it opens a mechanically gated channel on the sarcoplasmic reticulum. And if you remember from the first video, that's where we store a lot of calcium. So as a result, we have calcium coming in from the outside through the DHP channel, and then we have calcium going into the sarcoplasm from the sarcoplasmic reticulum. That calcium is important because it binds to troponin, which moves tropomyosin out of the way so myosin can bind to actin. And so at this point, we've got a cross bridge. Actin and myosin are bound together. And that's really important, but that's not all that we need. We need movement, and we need some other things that we'll talk about with our next video with sliding filament. Okay, so we just call this excitation-contraction. All right, so we need the excitation, we need that neural stimulus, that electrical stimulus to cause the contraction. All right, so at times we want this to stop, right? So how can we stop this process? Well, an easy way to think about this is that there are 10 steps, like I just listed 10 different things. All right, and so if we eliminate or stop or prevent or reduce or anything like that, we can keep actin and myosin separate, or at the very minimum, we can limit how much actin and myosin bind, or we can limit the number of cross bridges. So for example, if we go back to this picture here, okay, we've got acetylcholinesterase. That's an enzyme that breaks down acetylcholine. Now let's just say that we've got a lot of acetylcholinesterase floating around there, okay? So much so that it essentially eliminates or reduces or breaks, or however you want to think about it, the amount of acetylcholine. In fact, we'll just say it eliminates all of it or gets rid of all of it so that it can't bind to those receptors. Okay, and so as a result, we'll have reduced amount of acetylcholine binding. We'll reduce the amount of sodium that goes in. We'll prevent the action potential. Calcium stays in the sarcoplasmic reticulum, and really we're reducing the number of cross bridges. Okay, you got to remember, actin and myosin want to be together, but tropomyosin prevents this. Okay, now if you've had me before, I talk about how actin and myosin are like my daughter and her dumb boy. Okay, and just to be clear, I called him the dumb boy before I even knew he existed or knew who he was. Okay, and we're going on years now, so he's just always referred to in my house, and even by my daughter when she's talking to me, called the dumb boy, because that's what I called him 4 years ago when she was like 13. Okay, so she's actin, the boy is myosin, and then there's me, and I'm tropomyosin, okay? And to be perfectly clear, I hate it when I see parents do this. Like, I like to believe that I've raised good daughters, strong daughters that can take care of themselves kind of thing. Like, they don't need me walking around with a gun. And that would be scary anyway, because I don't know what I'd be doing with a gun. Anyway, in order to get tropomyosin out of the way, we need calcium, right? Calcium binds to troponin, which moves tropomyosin out of the way. When that happens, we have more cross bridges, right? So a stronger or more frequent stimulus that causes more action potential, or more action potentials leads to a greater release of calcium. This means we have more cross bridges. As a result, we have a stronger contraction. Well, how can we do this? Okay, and what's important, and this might seem counterintuitive, is we actually need to take calcium back up into the sarcoplasmic reticulum. Okay, and so you have to remember that calcium's leaving the sarcoplasmic reticulum via diffusion, and when it leaves via diffusion, it can go anywhere, right? It doesn't just, there's not a magnet that makes it so it goes to actin or troponin on the actin. It can go anywhere. It can go anywhere throughout that cell. We need it to go to troponin, but it doesn't necessarily go straight there. It just scatters, right? So just the example I've used is, just imagine that we have a big box of ping pong balls on top of the ceiling, or on the ceiling, and then we open it up. Where do those ping pong balls go when they drop? They can go anywhere. Okay, it's not like they're just going to go to one specific location. All right, calcium's the same way. Now, one of the things that will help make it more likely for calcium to bind to troponin is if we release more calcium from the sarcoplasmic reticulum. And in order for this to happen, we need to have a big gradient. So how do we create that big gradient? Well, the answer is with this right here. Okay, this thing called SERCA. And so what SERCA stands for is sarcoendoplasmic reticulum calcium. Okay, so we call it the SERCA pump. Okay, this is similar to that of the sodium potassium ATPase pump in that it involves active transport and it involves energy. And what this pump does is that this pump takes calcium that's in the sarcoplasm and pumps it into the sarcoplasmic reticulum. So that when a stimulus, an action potential, comes down, we open these doors, we've got a lot of calcium in here, reduced amount in the sarcoplasm, so we've got a bigger gradient. And when you have a bigger gradient, things move faster. Okay, and so when that ryanodine receptor opens, a lot of calcium leaves, or more calcium leaves. Okay, and so if we release more calcium, it's more likely that calcium is going to run into troponin, and if calcium runs into troponin, we form more cross bridges, and more cross bridges equals more force. Okay, and so in order to have a greater or a stronger contraction, we actually need to increase the amount of calcium that's in the sarcoplasmic reticulum prior to muscle contraction. Okay, and we'll try and work this out, act this out, something like that in class.